computer says it's 8:30. We have really one of my heroes today to present her work. I know Susan, she doesn't remember me because at the time when I met her, I was a student and had a very long hair almost to my shoulder. So I changed a little bit, and Susan did not. It is the same great scientist from the beginning. So there were really two people who discovered phosphorylation as a major regulatory way of cell to communicate and to transmit the signals and convert it into metabolic changes and so on. It was Krebs and Fisher. But the key enzyme that was studied was protein kinase A, and Susan is clearly a queen of this protein. Anybody working on protein kinases will learn what has been done on protein kinase A to get into the same kind of research in their own kinases. As we know, we have so many of them. So Susan, you always were my hero, and you are, and you're doing fantastic work, and we're so delighted to have you here. And let's just go to Sam.
Good morning, everyone. It's my great pleasure today to introduce Dr. Susan Taylor as part of our Distinguished Speakers series. She will be speaking today on protein kinase A signaling. Dr. Taylor is a professor in the departments of chemistry and biochemistry and pharmacology at the University of California, San Diego. She received her bachelor's in chemistry from the University of Wisconsin-Madison and went on to receive her PhD in physiological chemistry from the Johns Hopkins University in Baltimore, Maryland. After graduation, she completed 2 postdocs, first at the MRC Laboratory of Molecular Biology in Cambridge, England, and subsequently at UCSD, where she became a professor and has built a prolific research career, including as an HHMI investigator for 17 years. Her work has focused on the structure and biochemistry of protein kinases and how they contribute to signaling pathways within the cell. And one of her notable contributions include the first crystal structure of PKA. Dr. Taylor has published nearly 450 papers in illustrious journals such as Science, Nature, and Cell, and has been continually funded by the NIH since 1978. She has also been elected into the National Academy of Sciences, the Institute of Medicine, and the Institute of Inventors. Please join me in welcoming Dr. Taylor for what I'm sure will be a fascinating lecture.
Okay, so I should share my screen now. Okay. Can you hear me okay?
Yes, we can hear you.
Okay, great. Well, thank you, Sam, for that very nice introduction. And Christoph, I do remember when I first met you at the University of Washington, and one of the things you explained to me then very carefully was the importance of having membrane crystallography, of having structures of a membrane protein actually embedded in the membrane, and why that was so important. And I've never forgotten that. And even with a lot of the cryo-EM work going on, I still, that really important thing is how does that receptor interface with the membrane, because it's not just a neutral bed that it sits on. So I've always remembered that first meeting. So thank you so much for inviting me, and I will share with you several things, some history, but then I want to talk about some new things that I'm doing, in particular as a collaboration with UCI. And I think for me, it's really an exciting time. So I want to start out by really paying tribute to Roger Chen, who was my colleague for almost 30 years. We lost him tragically in 2016, but Roger, with his imaging of calcium and then his imaging of the GFP proteins, really showed us how we could use chemistry to understand biology. That was always the goal, to understand biology and how we could ask questions inside the cell. And my mantra has always been structure, and that, you know, for me, a structure is a starting point to understand function, but our end goal is still to understand biology. And now I think, especially these last few years, is disease. It's really providing a window into disease. And so those are kind of themes that have really resonated across my career. And Roger has made me always understand it's not enough just to have a structure. You need to understand what that molecule is doing in the cell. So that's one of the main themes I want to leave you with today. And so I'll go through and I will talk about some history for the first part, just PKA and what I've done most of my life. And then what did we miss? So this is a kind of very interesting thing. This the last 2 years really that this whole field has opened up again. And then the retina. It's serving for me as kind of a window into the brain, but it's also my window into biology. So I want to kind of share all that with you. And then the other theme is really how we go from molecules to cells to tissues, and why we need all of those things. That, and why in particular it's important to look at the tissue, what that cell does in the context of its neighbors. So those are two themes, understanding biology and disease, and then how we have to span this gamut. And so again, I'll go back to the history here. The field really began in around 1960 with Krebs and Fisher discovering phosphorylase kinase. And this was a regulatory kinase that phosphorylated and activated glycogen phosphorylase and let it break down glycogen to give glucose. And so it was a decade later that Krebs also discovered PKA. And it was actually a contaminant of the phosphorylase kinase prep. It actually phosphorylates phosphorylase kinase and activates phosphorylase kinase. So that concept of a cascade where one kinase phosphorylates another and another, that was actually there with those first two kinases. So that's one decade. So the next decade, there was all this flurry of activity around Src. Src was discovered to be an oncogene. I mean, that was a major discovery in the 70s. And then it was found to be a kinase, and then Tony Hunter showed it actually phosphorylated tyrosine. So that was going on way up there, the cancer biology people, whereas down below here, glycogen metabolism, how do we store and release energy? So my work has been focused on PKA, but it's served as a template really for the whole family. And this is like ancient history. My first graduate student, we knew biochemistry, we knew protein chemistry, and he did an affinity labeling experiment. This fluorosulfonyl benzoyl adenosine is an analog of ATP. At the end, it has a reactive group, and Mark Zoller showed that this modified one single lysine in PKA, is this lysine, and you protected with ATP, and when that was modified, the kinase was inactive. So those are pretty fundamental concepts. We didn't know even the sequence or anything. And so then in 1981, the Seattle group did the sequence. It was the first protein kinase to be sequenced by the classical way. It took like 5 years then, a long time. I actually got my tenure by sequencing lactate dehydrogenase. It's kind of hard to believe that. But, and then when we had the sequence, we knew this was lysine 72. And so one other key experiment, we knew there was a phosphate on this protein also there. And then in 1983, another graduate student, Joe Buechler, did a cross-linking experiment with a carbodiamide. So that crosses two primary amino groups. And he showed that two acidic groups, this 184 and 191, we could now place them in the sequence, actually cross-linked to this lysine. So we had this hypothesis that these three residues were somehow close to the active site, and in particular close to the ATP binding site. And so PKA wasn't actually first protein kinase to be sequenced. Src was. The family of these Src and Src-like kinases, cloning was coming into vogue about that time, the end of the 70s. And so they were cloned. The sequence, Src was cloned, I think, in 1979, but no one knew what a kinase should look like. So no one knew what to make of that sequence. And it wasn't until the PKA sequence came out that Margaret Dayhoff, she was a real pioneer in bioinformatics. She looked at that and she did all the sequence gazing. And even though there weren't a lot of homologies, she could align those and show that they were actually evolved from the same gene family. And so that was a really pivotal thing. That meant that whole kinome tree, they not just had kinase activity, but they all evolved from a common precursor protein. It was one big tree, and it's not like the phosphatases. They're all little trees and branches there. They're different mechanisms. These kinases all came from the same origin. And so that was the lysine 72. It was conserved, and that little peptide was our peptide. And the two conserved acidic residues that we found, they were also conserved. And so that was enough. At that time, there were maybe 25 or 30 kinases, many of them cloned, maybe 10, maybe not even that were sequenced traditionally, but that was enough for Tony Hunter and Steve Hanks to put together a family. This was pre-blast. And from that, they identified these subdomains, and it's still a very good definition of the secondary structure of these subdomains. And so for me, that's kind of the Rosetta Stone. It's the chemical definition of a kinase, but it doesn't tell you what's that lysine 72 doing in that structure. And so I had trained at the MRC lab in Cambridge. I learned protein chemistry side by side with crystallographers. And I really knew you had to have a structure if you were really going to understand that molecule. And so I started trying to crystallize this. And people kind of thought I was odd at the time because it was mostly pharmacologists, physiologists studying this, not crystallographers. So this was, for us, a really major thing to sit there that first day and see this first kinase structure. My two graduate students, Lynn Tenike worked with us at this time on refinement, and they each did a different refinement. We had no molecular replacement then. And so they did it separately, and then they got together to put their refinements together, and they wouldn't show it to me because they didn't want to bias their work by my trying to say, oh, I know where this should be. And so we all sat down together one day at the Supercomputer Center in San Diego and looked at this, and it was just kind of my really special moment in science. But what we've learned in between now is the dynamics. You can actually have the same structure, but it can have different dynamics. So here's that structure that we saw. And so what did it tell us? I mean, we didn't realize how almost perfect this structure was. It had a conserved fold. So we knew immediately all the kinases in that, what is now growing to be a very big family, were going to have the same fold. Those little white dots, those are the conserved residues, and they're like a little constellation. When you have an active kinase, they all will be superimposable in every kinase. There's lysine 72. Their ATP binding, we really knew the details of the ATP binding site 2 years later in 1993. And this was a novel ATP binding site. We didn't make enough of that at the time, but ATPases, hexokinases, they have the adenine ring of ATP sitting out on the surface with the phosphate in the middle of the cleft. This is a novel ATP binding site. Peptide binding, we also had this pseudosubstrate. It's an inhibitor, high affinity inhibitor that has an alanine at the P site instead of a serine. So we trapped it there. There are not many structures of a real substrate with an active kinase, even now. And the role of phosphates. So we have two phosphates there, this one in the C-terminal tail, and this one in what we're going to call the activation loop. And so this was kind of a lot. We didn't realize how much we learned from that. We thought, oh, they're all going to be so easy. But many kinases that we have now, they're inactive structures. And so my first thing was to look for those 3 residues. And of course, there they were right there at the active site with ATP. So it really, the essence of the active site was really captured perfectly by that affinity labeling. And I call this the conserved regulatory triad because it's there in every active kinase, along with those 2 metal ions. They're really critical. And we didn't appreciate how dynamic this active site is, but this is what the active site looks like. It's not transferring the phosphate. It's regulatory. It's positioning the phosphate to be transferred. And then I also want to tell you about that phosphate on the activation loop, because this was a completely active and ordered activation loop. And so if we look at that phosphate, it's not just an electrostatic node. It actually brings together all these parts of the molecule. And that kinase is very inactive when you don't have that phosphate there. So by putting a single phosphate on that activation loop, you really integrate the whole active site of the kinase. And so it's really quite remarkable what one phosphate can do. So this is like, I was doing another review recently and saying, so what did we learn in 1991 and where are we now? And all that we've learned in this time and 30 years. And so there's a lot that we missed, but I'm going to just tell you 2 things that I think are important. I told you first was finding ATP, putting ATP in that site. That was really important. Then this one, I'm going to tell you about this regulatory spine, and then I'm going to tell you about the C-terminal tails. And that's all. I'm not going to go into a lot of kinetics and structure details. So we learned a lot from PKA, but to really understand regulation, you needed to compare different kinases. You couldn't just learn this from one. And this was Alexander Kornev, who took a graph theory method and spatially compared active and inactive kinases. And from that, we discovered what we call the regulatory spine. And there were 4 residues that aligned. They weren't always the same residue, but they were always hydrophobic. These were always aligned in every active kinase. And 2 are in the N lobe and 2 are in the C lobe. And these are from motifs that we now know well from the kinase world. But that's a signature motif of every active kinase. And I'm going to tell you, PKA is assembled as an active kinase, and then it's packaged with an inhibitory regulatory subunit. But the tyrosine kinases like Src and EGF receptor and BRAF, they're assembled as a large inactive complex. And as part of the activation, you see the assembly of this spine. And so here's just a model. This is a morphing model now of Src. And you can see here's the lysine, here's the glutamic acid, and here's the glutamic acid. And you can see already the spine is broken. That's an inactive conformation. And if you now look at the morphing of this into an active one, you can see how the regulatory spine is assembled. And so if you look at that, you can see, first of all, that the spine is assembled, but you can look at this lysine and it's going to hook up with this glutamic acid. Those are my 2 residues. So you can see how it does that. When you assemble the spine, you make that contact. And then the other thing that you do, I didn't show on here the phosphate on the activation loop, but this activation loop is right here. And just watch it one more time. You can see how that activation loop now assumes a different conformation, and that phosphate is playing a role there. So this assembly of that spine is really important. It's a switch. And it's that dynamic regulatory triad again. So now, conserved core. So if you look at all kinases on that tree, the core looks pretty much the same. You can model an active kinase pretty easily. This is the core, the N lobe and the C lobe. So now, what about PKA? This is PKA and PKC. And we're doing a lot with PKC now as well, with Alexandra Newton at UCSD. But here's PKA, the full-length catalytic subunit with its tails wrapped around it. And if we look at just some examples of the kinase, here I told you about Src. It's got these domains here. There's a phosphate. This phosphate here hooks up to this, and that keeps it turned off. So as soon as you remove this phosphate, that's how you activate Src. But all of these kinases have these domains linked to them covalently. Even PKG, which is the closest homolog, has the cyclic nucleotide binding domains covalently linked to the kinase domain. And PKA is different. It's one little separate subunit. And if we look at the AGC kinases, these are just 3 of them. We see even this PKA catalytic subunit is pretty unusual. It's a complete, fully active kinase. And the regulatory domains are part of a separate protein. So that gives you lots of combinatorial diversity, which I'll talk about. You have different isoforms of R, you have different isoforms of C. And and they all have this conserved tail. And so what Kannan Natarajan, who was at postdoc in my lab now at the University of Georgia, showed was that this tail is actually conserved in all the AGC kinases. And so it's a highly regulatory feature for all the kinases. And in the case of PKA, you have this little N-terminal tail as well. And so I'm going to just show you, here's this core. By itself, it's really inactive. In many kinases that we just have the kinase domain without the regulatory domains. And you're missing a lot of the biology there and the biochemistry. Here's the movie of the tails. So this red one, that's a C-terminal tail, found in the C lobe and the N lobe. Here's the N-terminal tail, it's a long helix. And the other feature at the end of the N-terminal tail is this myrcellation site. So there's in the free C subunit, it's a acyl pocket that binds to, and but it can't be a membrane anchor, and I'll talk about that later. So here's just showing superimposed all these different AGC kinases here. And if you take away the kinase core, you can see how that tail is conserved. And this C-terminal part, we call it the C-lobe tether. It's always pretty much stable in all these kinases, whether they're active or whether they're inactive, and it's anchored to the C-lobe of the kinase. This end part here is anchored to the N lobe. So these are anchored. And this part can be completely disordered or ordered depending on whether ATP is bound. This is the active site tether, and this part will be disordered completely, or in our case, when we had ATP and PKI bound, that was fully ordered and contributing to the ATP binding site. Okay, so this is just a review that we did recently, which goes over a lot of the allostery and the dynamic regulation, and I just put it there. I'm not going to talk anymore about that. But I am going to now talk about the PKA Holoenzyme, and this is like a stone that my longtime colleague Shmuel Shaltil, who died, actually he died in the early 2000s, but really did a lot of the biochemistry of the catalytic subunit. And as soon as we saw that catalytic subunit, he gave me this and said, next you have to do the holoenzyme. And so that sits on my desk. I haven't been at my desk for since March now, but that stone sits there on my desk as a reminder of how important that holoenzyme was. So how's the activity of the C subunit regulated? So it's a different structure. There's a dimerization domain. That's the structure of the dimerization domain. And the red is an ACAP sequence. ACAPs are scaffold proteins that bind with high affinity, like a nanomolar affinity, to this domain and target PKA to membranes and transporters and things. And then it's followed by these two flexible linkers and then the two cyclic AMP binding domains. And so those are really classified as intrinsically disordered regions. And they have like an inhibitor site. They have an inhibitor site there that looks like a PKA substrate, and that's going to be important. But when you don't have C subunit, it's disordered. And so PKA in the cell is turned on by hormones, neurotransmitters binding to a GPCR, and then that transfers the message through G proteins to adenylate cyclase. The G proteins weren't even discovered when PKA was first discovered, and phosphokinase. But PKA sits there as an inactive holoenzyme in most cells, and binding of cyclic AMP unleashes the catalytic activity. But it's not just floating around freely. Most of it's anchored to specific sites, to mitochondria, to the plasma membrane, to Golgi. And so if we think about PKA signaling in a biological sense, we learn a lot from the free R and C subunits, but really physiologically, it's the holoenzyme that is really important, and that's a dimer. And it's there are 4 R subunits, and what's really important here from the genetics is they're functionally non-redundant, and I will tell you more about that as well. And then it's targeted as a complex, and so I call this like PKA signaling island. So even within a cell, you can have one PKA island that's at a transporter, another that's at the mitochondria, another that's by the nucleus. So little PKA signaling islands, and that PKA that's there is committed to that function, regulating that channel or that transporter. So just the R subunit, when we saw that structure, this one here, this inhibitor region was there, but it was disordered, and it had 2 cyclic AMP binding domains and was like CAP, the gene activator protein in E. coli. So it's a conserved cyclic AMP binding domain across all phyla. But it didn't tell us how was PKA inhibited, and so we needed to go to complexes. This is a complex of 1 domain and 2 cyclic AMP binding domains, and these showed us first how it interacted with the C lobe of the kinase, and then the little red part. Now you could see how that inhibitor site became ordered at the active site, which we predicted. But if you look at R1 alpha and R1 beta and R2 alpha and R2 beta, this structure here, they look pretty much the same. And so, so we needed to go to the holoenzyme, and I'm just showing you here one of the conformations of the R1 alpha, R1 beta. They're 95% identical in sequence. And the RC complex looks the same, but when you look here at the quaternary structure, you can see just from these movies, they're very different. And you can also see their phenotypes are different. You knock them out. R1 alpha is embryonically lethal, whereas R1 beta gives learning defects. So R1 alpha is really, it's the only one that's embryonically lethal of the R and C subunits, single ones. It's a major center of stress, of oxidative stress, energy state, metabolic state. It's a tissue-specific extinguisher of cyclic AMP-mediated gene expression. And most mutations, disease mutations associated with PKA are mutations in the R1 alpha subunit. Some of them, like Carney complex disease, create a holoenzyme that can be activated much easier. Others, like acrodyscystosis, they make it much harder to activate PKA. So there's a lot of genetic mutations there. But now let's go to the 4 of them. You can see they're all same domain organization. And you would think, you saw one, you saw them all. But I've already told you R1 alpha and R1 beta, when you look at the holoenzyme, they're really different. And so I'm going to go now to the R2 subunits. And these are almost always tightly bound to, through an ACAP to some kind of membrane protein, to a channel, to a transporter, to a receptor. And so they have some unusual features compared to, like, they have an inhibitor site too, but it's a substrate. Instead of PKI and R1 alpha, they're pseudosubstrate. They have that very high affinity binding with ATP. So they're very different, and that's a fundamentally different feature of R1 alpha subunits from R2 beta subunits. And so we started looking at the R2 subunits, and another mechanism that we use to study shape is small angle X-ray scattering. And this is the 3 R subunits, and you just need to look at this. This is the radius of gyration. It's how long is that molecule? What is the longest dimension? And you could see R1 alpha, R2 alpha and R2 beta are very extended, whereas R1 alpha is very compact. And so they're fundamentally different there. And Jessica Broyston's crystallized the full-length R1 alpha subunit and showed actually there's crosstalk between the 2 cyclic AMP binding domains, and it's very consistent with this compact structure, even though the dimerization domain is still very dynamic. To trap them all in a crystal structure is still challenging for R1 alpha. And then in this case, the R2 alpha and the R2 beta, those 2 RC complexes they don't talk to each other. They're more independent. So then we went to the holoenzymes, and we looked at R2 alpha and R2 beta when you add the C subunit. And now you can see R2 alpha stays very extended. It's not that much different than the free R subunit, but the R2 beta becomes very compact. It's the most compact of the 4 holoenzymes. And so we chose to focus on that for seeing, could we actually capture a tetrameric holoenzyme? And this is the RC complex. It looks pretty much like the one I showed you before for R1 alpha. 2 features I want to point out. This little alpha beta 4 beta 5 loop here, which is solvent exposed in the RC complex. And this little, we call it the FDDY motif. It's part of the ATP binding site, this phenylalanine and this tyrosine, even though it's in that C-terminal tail. So now they're just both exposed to solvent. And when you look at the holoenzyme, however, and this was solved by Ping Zhang in 2012, now you see this beautiful symmetry, which Jean-Pierre Changeau is always, he comes every year for 2 or 3 months to San Diego. This is the first time he won't come this next spring. And he's been telling me, you have to see the holoenzyme. You'll never understand allostery unless you see the full holoenzyme. And of course, here you see the beautiful symmetry that's in that holoenzyme. You can see how these 2 surfaces that were solvent exposed now are interfacing. This is RC and this is R prime C prime. So you can really see the allostery here, and it has a huge effect on activation. The RC complex activates at 60 nanomolar cyclic AMP. The Hill coefficient is 1. This tetramer, it's 10-fold higher. It's much more in a physiological range, 600 nanometer, and the Hill coefficient is now 1.8. So you really need the tetramer to capture the allostery. And I want to just show you, this is a paper that just came out a few months ago. This was a cryo-EM structure that we did in collaboration with Mike Cianfrocco, who's now at the University of Michigan. And although this is a little bit lower resolution, it allows you to see the shape of the holoenzyme. It shows that in this case, that linker region weaves through the middle of that hole where the symmetry was I told you about, and it pushes the dimerization domain below here. So, and it also shows in this structure that there's an intrinsic asymmetry between one side and the other side, that this can be very dynamic, one of these domains. And then it shows this is now poised to interact with an ACAP, but also the 2 tails of the C-subunit, which are myristylated, I told you that, they now are flexible in the R2 beta holoenzyme. They're still bound to the acyl pocket in the R1 alpha holoenzyme, but here they're flexible. And Ping also showed later that if you take the myristylated R2 beta holoenzyme, that can bind to membranes on its own. It doesn't even need the ACAP, whereas the free C-subunit doesn't and the R1 alpha holoenzyme doesn't. And so there's lots of ways of interacting with membranes here. This is a basic region following the myrcylation site that probably interacts with the acidic lipids. There's also this beta-4, beta-5 loop in the B domain is very basic. So one real challenge, Christoph, is to see what does this look like when it's anchored to a membrane? And I mean, that's an essential part of this story, I think. Anyway, we're getting there step by step. Okay, so this is just showing you how, and it isn't actually incorporating how that has to be at the membrane now, but just ACAP 75 or 79, ACAP 5 is its classic number, has all these motifs, and you can see binds PKC at this end, and here's calcineurin, and here's PKA, and PKA phosphorylates the tail of this voltage-gated calcium channel. And when it does, it opens it up for calcium, and calcium comes in, and calcium activates calcineurin. And calcineurin, now, we actually think this is a very tight complex between calcineurin and the holoenzyme. And this is anchored by an ACAP. So that's how you can think of an ACAP, and there's a membrane anchor here. So how is this whole big thing anchored to the membrane? It's going to be really an exciting challenge for me, not just looking at what does the channel look like when it's embedded in a membrane, but the next challenge, how do all these cytoplasmic things couple up and contribute to the regulation of that channel? And obviously this is going to take a lot of people, a lot of technologies. So what did we learn from Roger? What did I learn from Roger? Okay, you need to think about it, not just as your little kinase structure, but what is it doing in the cell? And how can we understand that? And so let's go back to these early studies. Stan McKnight cloned all of many of these different isoforms and did knockouts. And you could see R1 alpha, I told you, is embryonically lethal. R1 beta, you knock it out, you have learning defects. And R2 beta, you knock that out, you get a lean mice. They can eat all the high-fat diet they want. They don't get obese. They don't get resistant to insulin. So they're different phenotypes. And the alphas, C alpha, R1 alpha, R2 alpha, they're all expressed in most tissues and in constitutively expressed. But if you look at the beta isoforms, they're very rich in the neuronal tissues. And we focused especially on R1 beta and R2 beta, which are really highly expressed in the brain. And so again, you can see if I add all three of these holoenzyme structures, you can see they're all different. That R2 beta quaternary structure is different. And so Ronit Elouz, who was a postdoctoral fellow on my lab, focused on the R1 beta and R2 beta, which are highly enriched in the brain. And she did this high resolution, large scale mosaic imaging of the brain. And it's like you can do the imaging of the brain and then you can zoom in on particular regions. And you can see here, we predicted R1 beta because it's associated with learning would be rich in the hippocampus. And you can see that it is. I'm going to switch now to zoom in even further. This was an eLife paper that came out in 2017. This is what I want to do with the retina now, but you can see if you zoom in, you can see some regions have a lot of R1 beta expressed. It's very rich in the hippocampus, but then there are other regions that are really enriched in R2 beta. And you can see that they're functionally non-redundant, they're structurally non-redundant, and I think in terms of their localization, they're also non-redundant. And then you can zoom into specific cells, and then you can see like these little ones that do have a lot of R2 beta, that's Golgi. So R2 beta likes to go to Golgi. So you can see, even though it's not a strong green, it's R2 beta is there. And then you can move over to this mossy fiber region. I'm not a neurobiology person at all, but I'm learning. So you move over to this mossy fiber region where you did have a lot of R2 beta. And what Stan McKnight has shown is that this R2 beta that's enriched out here in these mossy fibers, it's anchored to an A cap. And when you remove that A cap, all of the R2 beta goes back to the cell body. So it's like so much biology that's embedded in this kind of approach. And why I think I told you in the beginning, it's important not just to look at your high resolution structure of your molecule, but to look at the cells and to look in particular at the tissues. And so again, this is sort of going from higher levels of complexity that allow you to appreciate the biology. But then you have these little islands, like at the mitochondria, these different PKA signaling islands even in the same cell. And so how do we, each level reveals a new understanding of biological function, and that you need to do all of this. Computationally with the atoms, you can do so much, but you need to go to the tissues and the animals. You need those phenotypes in the animals. So now I'm going to switch, and I'm, kind of told you PKA is the poster child for the protein kinase family, one of the largest gene families. So they started this initiative a couple of years ago, NIH, and it was called IDG, Illuminating the Druggable Genome. And they chose 3 families, kinases, GPCRs, and ion channels, to represent this concept. And so, like, here's the tree. This was 2009. There are thousands of crystal structures of kinases now, but they're really enriched in like BRAF. There's probably thousands of BRAF structures because they're so important for disease and for drug discovery. Many of those are highly enriched in the illuminated kinome. But if you go to the dark kinome, what are important kinases that are probably very biologically important, but haven't been looked at all in a structural sense? And so if you look at that, the very bottom of this dark kinome is this C beta. I mean, honestly, it's like, how could you think C beta could be part of the darkest part of the dark kinome? So let's go back to this again. Okay, so I told you about the R-subunits, but here's C beta. Look at it like the R1 beta and the R2 beta. It's very enriched in the neuronal tissues and in the brain. It's the forgotten isoforms. And so I think it really is a new frontier in PKA signaling. And you can look at how this is so enriched in the brain. Over 50% of the PKA in the brain is C beta, and nobody's really looked at it at the protein level. Are they biologically different? Are they targeted differently? And is C beta relevant for disease? So these are some of the questions. We got a little 1-year grant to look at C beta. And so just a kind of example of how my life goes. We just started this like a year ago last summer, 2018, July, we started. And then in August, Victor calls me. I didn't know Victor. He's in Spain. He's in Madrid. And he said, they have this very unusual phenotype of their patient. It says, he studies sonic hedgehog signaling. He said it gives kind of a lot of similarities to sonic hedgehog mutants. And he wondered if we were interested. So he brought together this team. Actually, Bjorn cloned a lot of the C beta isoforms, and Bjorn, myself, Fritz has done a lot of the biochemistry of this. And then there were these patients, several from Spain, from France, and then from Australia. So we had these conference calls for all of the fall and into the COVID sequestration. And we put this together. So 2 o'clock in the afternoon, San Diego time, you can reach all of these people on all parts of the world. And so this paper just got published at the end of the summer. And there were, these were the patients. So there were three different patients, 3 different families that had the same C alpha mutation. There were 4 different families that had different C beta mutations. And these are the ones that I'm going to focus on today. And they had these severe phenotypes. They had these polydactyl things. You get extra digits. That's a symbol of a sonic hedgehog signaling. So just some of these, they have many heart defects. They're very developmental kinds of defects. Skeletal abnormalities, short skeletal dental things. Ectodermal defects, cognitive deficits, and various unusual tumors, especially endocrine tumors, and all inhibit sonic hedgehog signaling when you put them into fibroblasts. So, okay, let's see. I'll tell you these mutations in the C beta. So there's 4. There's 2 at this histidine 88, and this one here, serine 54, and then there's a glycine down here. So I'll just tell you in general what they do. This one here, you can see this is the C helix. I didn't tell you very much, but you know, from that C helix is where that goes to my lysine, right in the back here. And this is part of the ATP binding site, and this is the glycine-rich loop. And so these really disturb. There's a histidine to arginine, a histidine to asparagine, and this is a serine to leucine. And this is that PKI peptide there. And so this disturbs ATP binding. And you can see this is just using a fluorescent polarization assay. This is the wild type C beta protein, and this is the His88 mutant. It doesn't bind ATP with that high affinity, which you really need for binding IP20. Here's IP20, the normal one, and you could see the mutant doesn't bind IP20 very well. And even the R1 alpha subunit, you can see is much easier to activate. So that's kind of a biochemical explanation for the consequences of those mutations. This mutation is in that hydrophobic groove that binds this amphipathic helix at PKI, and this is going to abolish binding of PKI or other things that use that groove. So 4 different mechanisms for giving this same phenotype. So, okay, look at C alpha and C beta. There's only 25 amino acids difference between them. I mean, you would think we expressed this 10 years ago and purified it and didn't pay much attention. You know, if you think, okay, that's not such a big difference. Okay, but if you look at those carefully, you can see that these are, half of them are in the N-terminal tail and the C-terminal tail. And I told you about that already. So you can see how those are part of the tail. These are regulatory mutation differences between C alpha and C beta. Here's the active site. There's no mutations at the active site of this. And so what do they do? So if we map it a little bit more, we could say, look at, there's these C alpha 1 and C beta 1 I showed you, but then there's all these other isoforms of C beta, and they differ simply by this N-terminal first exon. They have different, some is really short. C beta 4 has almost nothing. This has a long one. It has the glycine where it's myristylated. This is over 60 residues. This is about the same size, but it doesn't have a myristoylation site. So these ones, what I'm going to try and convince you that these are all functionally distinct. C alpha is different from C beta is different from C alpha, but the isoforms of C beta are also unique proteins, so that maybe we really expand this kinome even further. And it's not just 3 PKA isoforms that should be there, but in fact, a bunch of them. So that's what we're kind of exploring. And just these are the tails. If you just look at the tails, which I kind of told you about before, but I just think it's cool. Here, how this wraps around, N-terminal tail wraps around here, the C-terminal tail wraps around there. And we can think of these mutations as clusters. This cluster of, it regulates the N-terminus of the A helix and the beginning of the C-terminal tail. This one is where the very C-terminal part of the C-tail comes together with the A helix. And then this part is, the cluster 3 is really this part here, which now regulates this flexible linker. And just again, look at how we dissected that tail. And I told you before, this is a hydrophobic anchor in our structures, NPKC, all the AGCs, that's stably anchored to the N lobe. And this is always stably anchored to the C lobe. It stops right there. And so if we just look at this site, that's a variable region. When you don't have ATP and PKA, that's completely disordered, that red part. So we call it the active site tether. There's a bunch of acidic residues there. And there's how the C-terminal tail fits into that. And those are those little clusters that I told you about. So there's a ton of biology that's embedded in these seemingly few differences between C alpha and C beta. And so biochemically, we want to figure out what those are. But now look at tissue-specific expression. Okay, here's just a gene expression of the different, like C alpha, C beta. You can see R1 alpha, C alpha. They're expressed, like I told you, in every tissue. They're pretty much constitutively expressed. And what about C beta? So if you look at C beta, here it is expressed at low levels in a lot of cells, but it's really, you can see it's enriched in the brain, which is what we saw from the RNA hybridizations that of the embryos. And then if you look at tissue-specific expression and you look at, these are just C-beta, all the C-beta isoforms are there, but if you now dissect it into C-beta-4, you can see C-beta-4, that little one is highly enriched in the brain, but it's not in like C-beta-2, that very long one, that's enriched in lymphocytes, lymphatic tissue, spleen, thymus, but it's hardly at all here. Based on the RNA hybridizations, we don't see it very much in the brain. C-beta-1 is kind of the most constitutive. It's expressed at low levels in most tissues. So the tissue-specific expression is really important. It can be really important in one cell, but not in another. And these were the, again, the RNA hybridizations that Stan McKnight did. I didn't put this reference here. This is in the late 1990s. I'm just looking at C-beta-1, C-beta-4, C-beta-3 in the brain. And you could see that they're different. And they are different. And so I think this is really rich. We don't even know, what are these doing in the brain? And then again, the tissue specific expression is another thing. So what Dorota and we've been talking, I don't know, 3 years ago maybe, and she's been educating me about the retina and what a wonderful tissue this is to work with. And so we're exploring now, can we use the retina as a window into the brain? And it's also, can we use the retina as a window for Susan into biology? And so we want to look at the resting state, what normal state of the retina, but how is it affected by age? Parkinson's disease, Alzheimer's disease, diabetes. So it's just such a rich tissue. And this is one of the first experiments that we did with Dorota. And this is looking at C-beta. And you can see C-beta is highly enriched in this layer here, and then also in the retinal ganglia cells. So Dorota is really interested in the retinal ganglia cells. And in the beginning, that's where we were also really interested too, and very excited that C-beta was expressed there. So now you look at C-alpha. We'll just look at we have even better images now, but you can just see these two, they're very different. It's not in this layer here, and it's in the axons. You don't see any of it in the axons over here. And then if you look at the comparison, it's just to show you that C-alpha and C-beta are, I think, functionally non-redundant. We showed from the disease mutation it was going to be important for a disease phenotype. Here we show that these are certainly going to be functionally and non-redundant and targeted differently. And just to show you a close-up of this little region here, which even then we thought, oh, look at this. This is showing R2 alpha and that little region. And what is that? So that's these mitochondria, these ellipsoid bags of mitochondria in the cone cells. Here's the rod cells back there, and they also have mitochondria. And you can see R2 alpha is all around it, but R2 alpha is not in this bag of mitochondria, and R2 beta is not there, and C alpha is not there. So this is just like a really rich source of biology for the retina, for mitochondrial function. And Janae Rao is a postdoctoral fellow who's leading this project now. Frank Ma has done a lot of the imaging work with this, and this is just a work in progress I'm telling you about. So then Janae, because we thought, oh, this is now mitochondria, so to prove that, she did co-localization with an antibody that recognizes complex 4 of the mitochondria, and you could see how beautiful that overlay is. And so you can see, I just think these pictures are beautiful. I love to do pictures of molecules, but I think that these pictures of the cell are equally beautiful. And so you could see how enriched the C-beta is in this ellipsoid mitochondria in the cone cell. But even if you go down to the retinal ganglia cells where there wasn't much, you could see there's not as much there, but if you go down and you look at that in this, you can see that it does co-localize with the mitochondria. And the mitochondria here are much more like a normal cell. Here's the nucleus, and they're spread around in the cytoplasm. You don't have that bag of them like in the ellipsoid like you do in the cone cell. And this is just a close-up of this beautiful picture. I think this is so gorgeous. So this is really, for me now, okay, what about light and dark? What about fasting and feeding, aging, diabetes, Parkinson's disease, Alzheimer's disease? How does this change as a function of all those things? And even mouse and humans, you know, if you look at light, dark, and fast feed, those are coordinated in humans. We eat in the daytime when it's light, and we fast at night when it's dark. But the mice are opposite to that. So how do those cycles, the circadian cycle and the fast feed cycle, how do they differ with regard to these PKA signaling things? And PKA is known to be really important, both in fasting and feeding and in the circadian cycles. And so just to show you, this is for you, Christoph, to show you where rhodopsin is, because, you know, I thought this would all be up in those outer segments. This is rhodopsin, and you can see PKC alpha. There's a little bit of PKC alpha in some of those things there. But if you look at PKC beta, there's no PKC beta up there. And all those mitochondria are down here. So maybe you can tell me some more about this. I'm expecting to learn from you, Christoph, as well about what's going on here. And PKG is another big cyclic nucleotide signaling component here. And where is the PKG signaling going on? Is it here, or is it actually here in those outer segments? There's just so much biology here. And then this is looking at the R2 subunits, and these were images that Janae also did recently, just to compare R2 alpha and R2 beta. And you can see there are similarities. They do overlap to some extent, but there are differences also. And so you can see this, and we really need to do some more co-localization. R2 beta is a very good marker for Golgi. So a lot of the R2 beta is clustered around the nucleus. Sometimes, to me, it kind of almost looks like a polar distribution that's more signaling, more associated here with the axons than it is on this side, which is going to that mitochondrial bundle. But then you can see R2 alpha is expressed in every cell, and you can see here in those rod cells and things, it's expressed in all of those cells. So anyway, so the question is now, if we go back to, I've showed you C beta is in that, in this mitochondrial bundle here. So we know C alpha and C beta are, this is, again, R2 alpha, R2 beta, they're in R2 alpha all around there. It doesn't go into the mitochondria. This is C beta that's sitting there. But that doesn't tell us what C beta is that. Is that C beta 4? Is it C beta 3? Is it C beta 1? Which isoform is there? And this imaging doesn't tell us this. And we can't make antibodies to, can make antibodies to C beta 2, but it's very hard to make antibodies that distinguish the isoforms of C beta because there's such a small, it's just that N-terminal part. It's very hard to make antibodies to do that. So, so these are what we've learned. C alpha and C beta are localized differently. C beta is expressed in cone and rod cells. It's localized to the retinal ganglia cells also. R2 alpha and R2 beta are also localized differently. And C beta goes to mitochondria. And then what about these C beta isoforms? And so this is, again, something very new that we're just doing with Doroto using RNAscope. And she's been amazing in helping us to identify which, can we make RNAscope probes that will discriminate between these different C beta isoforms. And so these are the ones we have RNA probes to C alpha 1, C beta 1, C beta 2, C beta 3, C beta 4, and C beta 4 AB. And now they're just beginning to do this work with Dorota and my lab. And these are some of the results. We were trying to interpret these now, but you can see from here for the base scope that that Dorota did, and Frank is now trying to repeat these, that you can see that C beta 4 AB, these are the pink ones, they're different, and they are down here in the retinal ganglia cells. This is really nice. These retinal ganglia cells are all different, so we want to see which ones have R2 beta, and this looks like 4AB is the one that's mostly down there. If you look at C beta 3, C beta 1, it's they're here in these regions. You look also here, these are up in those cone cells that we were looking at up there. And then C-alpha-1, you can see C-alpha-1's in everything, like I told you, it's constitutive in every cell. So those are our first images. These were just really put together in the last 2 weeks. So it's kind of a very open-ended project. And I want to just say, I do think this is really a new frontier for PKA signaling, and it's opening up a lot of new biology, and it's also providing a window into disease phenotype. So I think there's huge opportunities here. And I think, you know, from my perspective, there's huge opportunities where our two campuses could build together with some of the resources that you have there, not only your wonderful ophthalmology department, but also Enrico Gertan, where you have the imaging center. He collaborates with a lot of people at UCSD. There's some really exciting ways we could work together, and especially get our young investigators building these new bridges to the future of biology. And so I want to thank everybody. I've gone, sort of as I've gone along, I've tried to thank people, but particularly I want to highlight Doroto, who's just been such a mentor to me in teaching me about, I mean, I still don't know nearly as much as I should about the retina. And I just want to thank everybody, and also to say to all of you, it's really, this is a wonderful window that it covers translational science, structural biology going to really translational science. So I thank you for your attention and for opening up this whole new world to me.
Thank you, Susan, so much. This was oh my gosh, so much work done over this year. So let's go to questions, and we'll start with Jim Hall.
That was really fun, especially for an ignorant biophysicist. I'm really glad you mentioned Roger, because I remember Roger going from the slide rule clip to his belt to the HP25 clip to his belt.
Absolutely, absolutely.
That was really great. But the question I had was, how much is the variety of ACAPs and what they're going to grab onto and what they're going to present to the kinases for phosphorylation? Because it seems to me that you sort of wind the cell up to regulate particular proteins,
right?
A particular cell. The example that I'm thinking of is the very parochial one of the protein that I worked on for a long time, aquaporin 0. And John Scott and Tamir Gonen showed that the complex of ACAP2, aquaporin 0, and PKA had a big effect on cataract. And I'm just wondering how the what the possibilities are for expanding the gamut of ACAP specificity are.
Yeah, I mean, that's really interesting. I didn't realize it was associated with cataract. So that also, you know, the combinatorial complexity of the whole thing, just even the PKA isoforms, and then you bring in the ACAPs, and PDEs are another big factor with that aquaporin ACAP that John has more recently been talking about. Kidney disease, it's really important in kidney disease. But yeah, that would be really important to look at. And I think, you know, we could look at it. Where is that ACAP there? We haven't looked at any ACAPs to see what's there. But I think it's really, again, important to look at the particulars in the particular tissues, because like the one I showed you, ACAP5, it's in the heart and it's in the brain. But you look at the heart, and most of ACAP5 in the heart is associated with R2 alpha, whereas in the brain, it's really associated with R2 beta. And so it really, those tissue things are really different. And so I would say, you know, I know John is focusing on the kidney, so we could say, what is it doing here in the retina, that ACAP and that particular PDE? Because when you bring a phosphodiesterase to this complex, now you break down the cyclic AMP, and so now you keep it in a phosphorylated state. It has an effect on the phosphorylation state as well. So it's another one if you have good antibodies. And I think bridging the biophysics, there's so much biophysics here. And so, you know, for you, like working with Enrique Grattan, I think you've already done some collaborations with them, doing the biophysics that he brings to the table with the biological functioning of the retina and looking at cataracts. I'm thinking blindness. I just heard a talk yesterday about a chemical that was targeting a spliceosome, and they had to stop the clinical trial because of blindness. That they got blind. So what's going on? What's it hitting in the eye that makes them go blind as a phenotype there? There's just so much to do. Yeah.
Okay, let's move to Corinne.
Thanks very much.
Yes, hi, this was an amazing talk, and Dorota, the histology is amazing. So looking at all the expression of PK in retina, my question to you is, in your patients, have you seen any vision or retinal dysfunction in the patient that had any of these mutations?
Well, that's kind of our next step and why it's so valuable to be with you, and also our Alzheimer's Disease Center at UCSC. So we have some Alzheimer's disease eyes that we're going to look at. We wanted to do the wild type first. We haven't yet looked at those. Can you see, like in a mild or severe Alzheimer patient, differences here? We don't know that yet. We also have the mouse model, and beta amyloid model, and a tau model for Alzheimer's disease, and we can look at those in the mouse. And so, being able to look at those samples, I don't know that. Dorota's looked at glaucoma. You have a pressure-induced glaucoma. Does that show a change in any of these regions? Now we know where to look at, and I just fasting and feeding. What happens if you look at fasting and feeding? Look at these this ellipsoid. There's so many things to do. So that's next on our agenda. We have those brains. We're trying to get some funding for this, but looking at those Alzheimer brain samples, eye samples, is going to be really important.
I meant the patients that you have in Spain and France and Australia.
Oh, no, I mean, these are live patients, so you can't look at
Right. No, I mean, do they have any visual?
That's exactly what I asked Victor.
Impairments.
That's what I just asked Victor last week when I was putting this all together, and I asked Victor, do they have visual problems? And he just asked, especially the patients in Australia, that's where most of the C beta ones are. And so I'll find out for that. I don't know. But even things like, Down syndrome. So I think all neurodegenerative diseases are kind of mitochondrial diseases, but Down syndrome, those have visual problems. Those Down syndrome patients have visual problems. And a lot of the other mutations, you look at major developmental problems and things like that, you don't get to really think about the eyes. What are the eye properties of a mouse that has an ACAP deficit? Are the eyes affected? They don't really do the visual screening. We're trying to do that with one of our colleagues at Iowa, where there's a ACAP defect in one of the main ACAPs that goes to mitochondria. Those are really good questions.
Thank you.
All right, let's move to Ben Lin.
Yeah, good talk. In fact, I have naive questions, and so how to publish the paper in the Science? Do you have any advice?
No, I'm not a good person to ask about that. I think it's important. I think it's horrible how it takes so long now to get 2 years to get a paper in Science or Nature or Cell. And the whole process, I think, is very flawed. And to me, it's important just to get the paper published in a public access journal. And I think, if it's an important paper, it will get cited, and that you want to get it out there, because even, if you wait 2 years, that's important information that's not out there. And then your ideas are, PLOS Biology is really good because it's a good online journal. eLife is a good online journal. And like Christoph, maybe you could comment on this. The crystallography community versus the cryo-EM community, I'm learning a lot about their different social differences. So the cryo-EM community is very positive about putting their paper on bioarchives right away. They submitted to a journal for the first time, and they put it on bioarchives so everybody can see it. And if you do that, then your data is there. You can talk with people and use your data. You don't have to wait 2 years to share it with people. Whereas crystallographers tend to not do it until it's accepted. And so there's a lot of things here. And I just think the whole idea that there should be so much pressure on young people to publish in Science, Cell, and Nature it's just done a lot of damage to the whole scientific community. So don't worry, we just get it out and
Susan,
publish.
a great comment. I think again, yeah, it's important for promotion and all of that, how people perceive, but it also introduces, I would say, corruption to science, because that's
Absolutely.
not conscionable to keep papers for 2 years before they published.
Right.
Susan, I have 2 questions to you. So there must be a lot of mice with LOXP on those subunits. That could be quite useful to look on the function of the retina, which is so accessible to physiological recordings.
Yeah.
And so is there like all of those subunits right now, they are flagged so we can be removed tissue specific and maybe even inducible way?
That's, I mean, some of them have been, but some of them haven't. I mean, we haven't done those experiments with the PKA knockouts at all. And we have like, I mean, this would be a wonderful grant, just a foundational grant to do, and to do it also with Enrico, because you can make out each of those selectively. And like my colleague in Norway, Bjorn Skolhag, who works on C-beta 2, which is enriched in lymphocytes. And I think personally, I think it's going to be an indicator for inflammation in any tissue, but he can do a specific knockout of C-beta 2. And if we find that like C-beta 4AB, you know, I don't know, Gerardo, if we can even do this, if it's C-beta 4AB that's in the mitochondria, can we specifically knock that out? I mean, but we have an HEC293 cells, one knockout of R2 alpha, R2 beta, and another with R1 alpha, R1 beta, but we haven't even done anything yet with specific knockouts of the C-beta isoforms.
And the retina will offer you that you can take from particular cell type.
Absolutely, absolutely.
That would be really gorgeous, because again, you may know we will have an electrophysiologist joining our group, Vladimir Kefalov, who did some of the work on phosphatases. So this will be also in line of his interest. He should be here in May. So that's wonderful.
Where is he coming from?
From WashU.
Okay. I would love to do that. And it seems like this is just such a logical thing to put together in a big multi-PI grant. It's just,
You know that I hold one question to you from a long time ago, since you know that long hair I had.
I don't remember the long hair. I remember.
So let me then ask this question that always bothered me. I didn't get a proper satisfaction. So the PKA has a very defined consensus sequence for phosphorylation. As you know, Kemptide, the peptide, Bruce Kemptide did passed many years ago. How many, if they are, exception to the rules of this consensus sequence that PKA can phosphorylate in biology?
Okay, that's a very good question. And I don't think that has been systematically looked at. And I think it's, for me, a really important question, because it's not so important if you have it all tethered together and the tail of the channel is sitting right there by the kinase. It doesn't really matter that you have that precise sequence motif. So one of the most interesting things that I've seen recently in the last year, not less than a year, is Knepper. He studies kidney at the NIH, and he did a knockout of C alpha and C beta in kidney proximal tubules. And then he looked at, you know, what proteins are up or down regulated when you knock them both out, but what's up or down regulated, and what proteins are phosphorylated and dephosphorylated when you knock out each one separately. And he found something that I thought was really interesting, but it hasn't been a systematic looking at this, that you found different proteins were up and down regulated, but he also found that the substrates were also different. The phosphorylated proteins also had a different recognition motif. So the ones that were associated with C alpha, all were the classic ones, the arginines, and they're all classic ones. But the ones that were gone when you knocked out C beta were not classic ones. They had like a histidine on the C terminal side. And so I think there will be a big difference in recognition, but nobody's very much looked at those families of proteins that don't fall. There's a lot of well-known PKA substrates that don't have the classic motifs, but people go for the classic ones just because those are easy to identify. But I don't think even in the databases on phosphosites that there's any been systematic analysis of those known PKA substrates that don't have a classic site? That's still a good question.
Let's move on to Chris Sander.
Hi there, Professor. I'm a student in Professor Palczewski's lab. I have a question about PKC in the retina. I noticed in one of your slides that I didn't see expression of phosphokinase C in the retina, and it brought my mind back to a paper on arrestin translocation being instigated by PKC agonism. And so I was wondering if maybe the type of staining you were doing was for a particular isoform or something like that, or what you think is going on there.
Well, so you didn't, I didn't point it out, and you didn't look close enough, but on all of the images of the retina that we did, we used PKC alpha as a common reference standard for the synapses. So if you look at, if you go back and look at those, you can see, I don't know if I have it. Where is the PKC alpha? Yeah, here's a PKC alpha as a marker for the synapses. So that's on all of them. Then you can compare them one to another. And so they're at the synapse here and down here. It's not up there. But the PKC isoforms are the same. I mean, Alexandra Newton in my lab studies those. I know, like PKC delta, you have an ataxia. There are different phenotypes associated with those. But PKC alpha is a marker for the synapses. Hiroto could comment more on that, but that's the one PKC isoform that we looked at, and we're just starting to look at PKG to see where PKG is. But yeah.
Yeah, but if I may comment exactly, Chris, that was exactly the question we want to also think about, because those PKA is surprisingly now, I mean, for us it was surprising that it's in all over the photoreceptors. So this can be exactly one of the enzymes regulating translocation of different proteins during the visual cycle, day and light cycle. So it's one of the interesting questions, as we said, day and light and circadian rhythm. That's something we still don't know.
Is Professor Weiss online?
Thank you.
Greg, are you there?
Yes, I'm here. I've been enjoying this enormously. Hi, Susan. It's great to hear your beautiful work.
Greg, so I give you a chance to make a final comments today.
Oh, well, the thing that really struck me, and thank you very much for this opportunity. The thing that really struck me is something that I've always appreciated from Susan's talks, which is the scholarship and also the incredible focus over a long period of time. Those two things together, just to combine to build a really rich story that has these narrative arcs that go over the course of decades, and in doing so, just continually turn up rich veins of new information and fascinating, exciting new directions for the field to go. And I find that so exciting, Susan, and I'm just so thrilled to hear the latest from you. Thank you so much for enriching us today with this beautiful talk, and as always, sharing just fantastic science with us.
Thank you, Greg, so much. Thank you, Susan. Again, don't be